G'day, my name is Nigel Todd. I'm from Perth Region NRM, and I'm here today with Dr. Andrew Werrett from Living Farm. We're here in the Perth Hills at one of our nitrous oxide emission sites. The farm around us is a pear, apple and avocado orchard, and we're here to do our last trial on one of our sites. The project is a government funded project through the Action on the Ground program, and the project is aimed at measuring nitrous oxide emissions through horticultural enterprises. Andrew, would you mind sort of taking us through the importance of nitrous oxide emissions and providing us a link between nitrogen-based fertilisers, the nitrogen cycle and greenhouse gas emissions?
Yeah, no worries. Nitrogen obviously is, well, from a production perspective, is a vitally important, the most important nutrient. So we're of course interested in using it in the most efficient way possible, but recently it's become more topical because it's a very significant greenhouse gas. It's 300 times more damaging than carbon dioxide. So for every one molecule of nitrous oxide out there, it's does 300 times as much damage. Or you need 300 times of carbon dioxide molecules to do the same amount of damage. So it's important. A little bit can have play a big role. So what we want to do is, from an efficiency perspective, look at nitrous oxide, because that being produced from the soil is a loss. So from a production perspective, it means you're not using your nitrogen as efficiently as you could do, but it also means there's potential there to harm the atmosphere and add to climate change. So that's why it's important to acknowledge it and then also try to measure it and see if we can do something to mediate the effect that it has on the environment. You can't totally rule out this. It's a natural process. If there's nitrogen in the soil and there's moisture, you will produce nitrous oxide, but you can manage it, or hopefully manage it in such ways that you can minimise that loss. So that gives more back to the trees or the crop and less to the atmosphere. And certainly there are some conditions that help it. Waterlogged soils, overuse of nitrogen, having the excess amount of nitrate in the soil will lead towards denitrification, which is the process that then produces nitrous oxide. So certainly that's what we're trying to do here today, and we have been doing with this project, is use these soil ameliorants and trying to see if they have an effect on nitrous oxide production.
Well, as you mentioned, we're trialling a number of different soil amelioration products with the hope that we might be able to identify one or more that might be able to help us reduce the nitrous oxide emissions. Andrew, can you take us through how we are conducting this trial today and how we're applying the different soil amelioration products?
Yeah, no worries, Nigel. The important thing here is that we're using the farmer's existing system. So we're using his irrigation, and essentially all we've done is plug into one end. We've taken over control of it, so we're applying all the water and the nitrogen, and then we're putting in 4 different soil ameliorants, if you like, and seeing what happens. So the system here uses a gas injection system that we use in our small plot field research, which just involves using a small amount of LP gas pumped in through a bottle, and in each of these bottles is the mixed up product, and then you've got a trigger out further down. And that will open the valve, if you like, and pump it in through this injection coupling that you see now. This has a little orifice plate in it to regulate flow and provide a bit of feedback. But we can monitor the pressure here, and knowing that it's a bit more than the pressure in the line, we know that we're going to be putting product through the line. The important thing with this type of research, and particularly whenever you're dealing with soil biology and soil microbes and anything that they may produce, is that they're influenced by many outside factors. So soil condition, nutritional status, things like pH, soil type, all these things are influencing factors. So the more you can minimise variation in those factors, the better it is, particularly for greenhouse gas emissions. So what we've tried to do is we've got, even though there's 5 rows side by side, we can't really randomise the treatment because we randomise each of the chambers because we're using an existing tree setup, so there's rows of trees and the irrigation underneath it. So we need to tap into each of those. But what we can do is, if we can't limit the variation, is we can measure it. So measuring things like soil temperature, knowing what your soil type is, and knowing what the air temperature, soil moisture is, all contributes to understanding. And so we can use that for figuring out exactly what the influencing factors are and deciding whether it's these products or not that are affecting the way the soil microbes work. If you want to, we can go and have a look at one of the chambers. What we can do is we've also got these little, we'll use these in a while to check that our flow rates are right. But you can hear the pump going. So you can see under here, this is one of the chambers, and we've just sat the irrigation up on top of there. We've used this string, it's a little bit agricultural, but that's where we are. But that just ensures that the drip goes into the centre of the chamber and doesn't run along the tube. So we try to, you can actually see the colour is a little bit brown there, and that says that that tells us that the product's coming through. So it'll take us about an hour to get all of the products out. We're not too concerned with how long it takes. We want to reflect, these guys water for about an hour to an hour and a half, 2 hours at a time. So we're trying to match that. All we want to do is make sure that we get the exact amount of product out completely. So we've applied the recommended rate of each of the products out on a per hectare basis. So you can see one of the other things we've done. So this is one of the lids, that'll go back on top of there, is we've got a little soil temperature probe. So this is measuring air temperature and soil temperature in the irrigated zone, somewhere along here at about 5 to 7 centimetres, which is where you'd expect the microbes to be operating, and they're highly dependent on soil temperature for their activity, soil temperature and soil moisture. So we'll know those, and we can use that to see if there's any other influencing factors.